Method in biology, molecular biology and recombinant DNA method, isolation and purification of nucleic acid, RNA and DNA. The major components of cellular metabolism are nucleated as they constitute the nucleic acid, the deoxyribonucleic acid, DNA, and ribonucleic acid, RNA, which acts as the major component of information storage and transmission at a cellular level. Thus, for basic investigation regarding the molecular processes within a cell, isolation and purification of these nucleic acid is of high importance. A major hindrance is this in this isolation procedure is the mixture of different cellular components like protein that contaminates the nucleic acid mixture. The three basic steps involved in the isolation of nucleic acid are cell lysis to removal of contaminant like protein, salts, DNAs or RNAs. 3. Recovery of DNA or RNA molecules. Cell lysis. The cell lysis or disruption of cell membrane can be achieved in several ways, including the chemicals, mechanical and enzymatic methods. Chemical disruption, osmotic lysis. Osmotic lysis is one of the simpler form of chemical lysis that are employed. In this procedure, cells are usually suspended in a hypotonic extracellular solution, which is most common a solution of sucrose. This leads to osmotic diffusion of water across the cell membrane that eventually leads to rupturing of the cells. 2. Organic solvents like alcohol, ether and chloroform disrupts the cell wall present in plant cell by increasing the permeability of cell wall and cell membrane. 3. Ethylene diamine tetraestic acid EDTA, a known chelating agent, is widely used in the lysis of gram-negative bacteria whose cell wall contains lipopolysaccharides that are stabilized by the cations like Mg2 plus and Ca2 plus. The chelation of these ions by EDTA leads to disruption of lipopolysaccharide layers, causing opening within the cell membrane, leading to lysis. 4. Surfactants. The surfactants disrupts the ionic interaction between the hydrophobic and hydrophilic portion of lipid surface of a cell membrane, causing their lysis. The surfactants, also known as detergents, are widely used components of cell lysis buffers that solubilize membrane proteins and lipids, thus leading to cell membrane disruptions. Most widely used surfactants are tritone and sodium dodecyl sulfate SDS. 5. Catropic agent. Catropic agents like urea and gonidine are used for cell lysis. These agents can bring some hydrophobic components into the aqueous components of the cell. This is achieved by disturbing the surface of the water and making it a less hydrophilic environment and weakening the hydrophobic interaction among solute molecules. Mechanical disruptions. 1. Mortaring. The mortaring is a simpler method used for disruption of cell membrane by using the mechanical grinding with mortar and a pestle. This is most commonly done in case of samples frozen in liquid nitrogen. After cellular disruption, the resulting released component are collected in solutions. 2. Bead beating. Bead beating is a method where glass or ceramic beads are added in a suspension of cells and vortex or spun at higher speed to break down the cells by forceful grinding. 3. Sonications. Sonication involving sonication frequency to disrupt the cell membrane. So ultrasonic homogenizer functions by generating the vibrations in a titanium rod, which is then inserted in a cellular solution. The rupturing of the cell membrane happens via a process called cavitations, in which small bubbles are formed, which then burst, causing the localized shock waves and change in pressure. 5. 5. Homogenizer. The homogenizer where shearing force by squeezing cells through a tube that is slightly smaller than the cell diameter, thereby shearing away the cell membrane during the process. This process can also be achieved by using the rotating blade as in a blender. 6. The freeze thawing is a procedure in which the cells are first freeze in liquid nitrogen as minus 195.79 degrees centigrade, which leads to formation of ice crystals within the cell that expands in volume during thawing, thus rupturing the cell membrane and releasing the inner components. 7. The high temperature and pressure disrupts bond within the cell membrane along the protein denaturation, which is required to remove the histone proteins present alongside DNA within the nucleus. Enzymatic disruptions. Naturally occurring enzymes are used to remove the cell wall and release the cytoplasmic content. The most commonly used enzymes are lysozymes that disrupts the peptidoglycan layer of the cell membrane, cellulase, chitinase, mannose, glycans, etc. Removal of contaminants. Upon lysis of cell membrane, the least nucleic acid remains in a mix with other intracellular compounds like protein being present along with certain enzymes that degrade nucleic acid. The removal of these contaminant is thus important for producing a pure and stable environment extractions of DNA and RNA. Some main processes of this step comprises of 1. Enzymatic treatment of lysed cellular compounds used. Proteinase K is a most potent method of removing protein components by degrading them. The protein K is a broad spectrum serine protease that digests contaminant protein. 2. The proteinase K is highly effective in isolating DNA or RNA in undamaged native stage. In the presence of 0.5 to 1% SDS, the purification of genomic DNA from microbial culture is a standard molecular biological experiment. Involves the cellular lysate by treating with proteinase K at 37 degrees centigrade for 1 hour in presence of SDS. The reaction SDS acts as a denaturizing agent which unfolds the protein structure, allowing greater digestion by proteinase K. 3. The proteinase K is stable up to a temperature of 55 degrees centigrade and thus increasing the temperature of the reaction at 50 degrees centigrade, increasing the digestion efficiency of proteinase K as more protein gets unfolded at a higher temperature. 5. A major functions of protein K is to degrade a different proteinase that degrade DNA and RNA and thus stabilizes the isolated nucleic acid in a solution. The proteinase K. Proteinase K is a broad spectrum serine protease discovered in 1974 from fungal extract of Gyodontium album. The K is named is derived from its ability to digest the protein keratin. The proteinase K has a molecular weight of 28.9 kilodalton and its cleavage and cleaves it substrate at the peptide bond adjacent to the carboxyl group of aliphatic and aromatic amino acids with blocked alpha amino group. The enzyme is stabilized by the presence of Ca2 plus ions, which does not contribute to the enzymatic activity but towards its stability. Another major steps towards the removal of protein components from the cellular extract in purifying DNA is achieved by extraction with phenol. 1. DNA and RNA are both polar molecules due to the presence of negative charge in phosphate backbone. This makes nucleic acid very soluble in water while being much less soluble in phenol. In the phenol extraction procedure, the phenol is thus added to the cellular extract in equal amount and then water excess and mixed. Following centrifugation, the much heavier phenolic compounds containing protein flocculation is present in the bottom layer while the nucleic acid remains in the aqueous layer lying above it. 2. The proteins are composed of amino acids characterized by the presence of both polar and non-polar group. Under normal cellular conditions, the polar hydrophobic side chains of proteins remains exposed to the cytoplasm, while the non-polar hydrophobic groups remains hidden within the inner fold of the molecule. However, in the presence of phenol in solutions, the localization of these side chains is reversed, with the non-polar group being exposed to the exterior and thus becoming soluble in phenol. 3. In laboratory method, the phenol extraction is performed using a mixture of phenol, chloroform, iso amyl alcohol 25 is to 24 is to 1. As the chloroform removes the lipids from the solution, leads to the greater denaturation of protein, allowing greater dissolution of a protein and the removal from the aqueous layer. 3. Recovery of DNA or RNA. DNA precipitation. 1. Following the removal of contamination using the phenol solution, nucleic acid is precipitated from the aqueous solution with ethanol. During this step, usually the sodium acetate pH 5 to 5.5 is added with the function in neutralizing the negativity charged phosphate backbone, which promotes hydrophobic interaction, leading to precipitation. 2. The precipitated nucleic acid is concentrated by centrifugation and then removed or removal of supernatant ethanol solution. 3. The precipitator is then rinsed again with 70% ethanol to remove any salt present in the nucleic acid. 4. Following this, precipitator is dried to remove any trace of ethanol, as it may cause problem in further downstream processing. 5. The DNA or RNA precipitation is then dissolved in certain buffer solution that inhibits their degradation and is kept under low temperature conditions for long-term storage. RNA isolation. The extract of RNA is much more complication than the isolation of DNA due to the wide presence of RNAs. These RNAs are much more soluble, is much more stable than DNAs that are present. Therefore, the RNA isolation is proper form requires that the procedure to free from any such RNA contamination. This is done by taking extreme care during the different stages of RNA isolation. The different isolation agents are prepared using the diethyl pyrocarbonate DEPC treated water, which is free of RNA contamination. The washing of different apparatus is also performed using the DEPC water. Breakdown of cells for RNA isolation and stabilization of RNAs performed by using the guanidine thiocyanate, which acts as a catotropic agent in destabilizing RNAs. Following this step, RNA isolation is performed following phenol extraction. RNA precipitation. 1. Apart from precipitation with ethanol and sodium acetate salt, another more efficient method for precipitation of RNA is the use of lithium chloride LiCl. The lithium chloride precipitation offers major advantages over other RNA precipitation method in that it does not efficiently precipitate DNA, the protein or carbohydrate. Thus, the RNA precipitate is free of any contaminants and does not require any further purification. 2. The procedure is like ethanol precipitation of DNA and involves equal volume of lithium chloride added to the aqueous solution after phenol extraction, which is then incubated at minus 20 degrees centigrade for significant amount of time to allow precipitation, followed by the centrifugation to concentrate the precipitate. 3. The major factors in RNA precipitation using the lithium chloride are A. The temperature in which the precipitation can happen, 2. The concentration of lithium used, and 3. The duration and speed of centrifugation used during the final concentration. Isolation and purification of a protein. The proteins are functional building block of cells and are involved in almost all functions performed at a cellular level. Therefore, isolation of proteins and understanding their specific role is essential to a molecular investigation. The major steps involved in the isolation and purification of protein can be summarized to 5 basic steps. A. Extraction of a protein component from biological samples. B. The separation of non-protein components like DNA, RNA, lipids from extract. 3. Precipitation of bulk protein from crude extract. 4. Use of chromatographic methods to separate the protein fraction from the crude extract. Further purification to purify the target protein for using the finer chromatographic method. Extraction of protein. For extraction of protein components from a cell, complete disruptions of cell and release of compound contents from cellular debris is a major step. This process is mostly performed at a temperature of 4 degrees centigrade in order to avoid any degradation of protein. During the process, two important reagents are EDT and alpha mercaptoethanol. The EDTA serves to inhibit protease actions, and alpha mercaptoethanol is necessary for maintenance of a reducing environment. After the addition of the reagent in presence of a buffer solution, the mixture is homogenized and centrifuged to remove the cellular debris. The pellet is then discarded while the extracted protein remains in the supernatant solutions. The other four stages are more achieved by a combined effort of different chromatographic methods, which are detailed as follows. Column chromatography. The column chromatography is one of the most powerful method of fractionation protein. This process utilizes the difference in protein charge, size, binding properties, and other chemical properties of the protein. The basic methodology of a column chromatography involves a stationary phase which is composed of porous solid materials having appropriate properties of isolating the target protein or protein fractions, and a mobile phase which is a buffered solution that percolates through the solid matrix. The protein containing solution is layered on the top of this column, which then passes through the column, while individual proteins present within the solution migrates differently through the column based on their chemical properties. Based on the different types of properties used, column chromatography can be of many different types. Here, cation exchange chromatography. In this type chromatography, the solid matrix is characterized by having negatively charged groups. The protein have higher positively charged amino acid groups, thus show down while passing through this solid matrix, while the negative charged protein passes through rapidly. The two types of protein separates into two distinct phases as they passes through the column. The expansion of the protein band in the mobile phase, the protein solution is caused by separation of protein with different properties and by diffusional spreading. As the length of the column increases, the resolution of two types of proteins with different net charge generally improves. However, the time required for this process to complete can also lowers the resolution by diffusional spreading of protein band. B. Size exclusion chromatography. This type of chromatography separates the protein according to the size. In this method, the large size proteins appears first from the column, while the smaller size protein appears later on the column. This is achieved by constructing the solid phase with beads having engineered pore of definitive size. This leads to protein having diameter greater than the pores to pass through the liquid phase, while the protein having smaller sizes ends the beads and gets slowed down as they move through samples the solid phase. C. Affinity chromatography. The affinity chromatography is based on binding affinity of proteins with the group covalently attached to the beads, forming the stationary phase of the column. A protein that has a chemical affinity towards this attached group will binds with them and will be slowly down. The attachment can be later removed by elution through chemical agents that weakens this attraction or break the covalent bond between the attached group and the solid phase. For example, certain proteins are purified by expressing an oligohistidine tail to their carboxy terminal end. These proteins can be purified by affinity chromatography on a nickel resin due to histidine attachment to nickel group. D. A high performance liquid chromatography. The high performance liquid chromatography HPLC is a more modern approach to column chromatography. HPLC procedure utilizes a high pressure pump that increases the movement of the protein molecules down the column. They are characterized by high quality stationary phase material that can withstand the increased pressure generated by the pump. The use of high pressure reduces the transient time of protein through the column, thus minimizing the diffusional loss of resolution in protein purification. The analyzing of DNA, RNA, and protein. The extraction and isolation of nucleic acid and proteins are essential first step in initiating the molecular investigation. These steps are followed by the initial quality check of isolated products, followed by further analysis. The different methods of such analysis are detailed in the following sections. Spectrophotometry. The biomolecules like DNA, RNA, and protein can be observed light at specific wavelength. The principles of light absorption is used in spectrophotometer in order to quantify the extracted biomolecules and to get a qualitative understanding of the purity of extracted product. The spectrophotometer analysis of biomolecules utilizes the principles of Lambert's-Beer's law, which says that the fraction of incident light observed by a solution is related to the path length of the observing solution layer and the concentration of observing biomolecules. Log I0 divided by I is equal to delta CL, where I0 is the intensity of the incident light, I am the intensity of the transmitted light, and the delta is the and the sigma and the delta is the molecular extinction coefficient. C is the concentration of the observed molecules. I is the path length of the sample. The Lambert's Beer law assumes that the incident light is parallel and monochromatic, that is, having a single wavelength of the log I0 by I, and termed as absorbance A. The molecular extension of coefficient varies from solution to solution for each different biomolecules. The absorption maxima, that is where the maximum absorption of incident light occurs for both DNA and RNA, lies at 260 nanometer nm, with the nitrogen base being the major absorptive group of molecules. In the case of protein, the absorption maxima lies at 280 nanometer, with tyrosine and tryptophan being the major side chain components that contributes to the absorption. Thus, in an extracted solution, the purity of nucleic acid is checked by observing the ratio between the absorbance value at say 260 nanometer and 280 nanometer. The reverse is done in case of checking the quality of protein isolation. Gel electrophoresis. The protein can be separated by differential migration of protein in an electrical field in a method known as gel electrophoresis. The same technique is used for isolation of DNA and RNA also, but with certain differences. The protein purification using gel electrophoresis. 1. Separation of the protein using the electrophoretic method is carried out in a gel matrix made up of cross-linked polymer polyacrylamide. The gel matrix acts as a molecular sieve that slows down the migration of the protein approximately in the proportion to their charges to mass ratio. The shape of the protein also plays a part in the speed of the protein migration that through the gel electrophoresis field. 2. The speed of the protein molecules, that is its electrophoretic moiety mu, is calculated as the ratio between the electrical potential E of the protein and the velocity V of the molecules in the electric field. Mu is also expressed as a ratio between the net charge of the molecules Z and the fractional coefficient F generated due to the protein shape. Mu is equal to capital E divided by capital V is equal to Z bar F. 3. Another major component of polyacrylamide gel electrophoresis is addition of detergent solution dodecyl sulfate SDS, which binds to all the proteins roughly proportional to their molecular weight, with about 1 molecules of SDS getting attached per 2 amino acids. The negative charge present in the SDS makes the negative charge of the protein insignificant and gives each protein a similar charge to mass ratio. 4. The SDS binding also alters the conformation of proteins, making them assumed a similar shape. Thus, electrophoresis in presence of SDS separates the protein exclusively based on the molecular weight. The larger sized protein have a greater negative charge and passes through the electric field slower than the smaller size protein. 5. Following electrophoresis, the entire gel matrix is treated with a dye to visualize the different protein components that have been separated based on their size. One of the most widely used dye is Comasi blue, which binds to the protein but not to the gel matrix itself. The dye allows the visualization of the protein separated based on their size. If multiple subunits of proteins are present in the solution, then each subunit gets separated based on their size and get visualized as separate bands in the gel. Isoelectric focusing. The isoelectric point pi of a protein refers to a pH in the surrounding where the charge of the protein becomes entirely neutral. The isoelectric focusing is a method used in gel electrophoresis that utilizes this property of a protein for better separation. In this process, the pH gradient is established by allowing a mixture of low molecular weight organic acids and base to distribute themselves in an electric field generated within the gel matrix. When a protein mixture sample is added, each protein travels up to the pH where its net electric charge becomes neutral, that is its pi. Thus the protein that have different pi gets separated by using the isoelectric focusing. Two-dimensional 2D gel electrophoresis. The two-dimensional polyacrylamide gel electrophoresis is a procedure that is applied to complex protein mixture extracted from cellular and tissue extract. The method is a combination of isoelectric focusing that acts as the final dimension of protein separation based on pH gradient, which is followed by another SDS phase that introduces a second dimension which separates the protein based on molecular masses. In a typical 2D phase, the isoelectric focusing is performed in a tube gel which is cast within the thin plastic tube with a pH gradient existing along the length of the tube. 2. After running the isoelectric focusing, the tube gel is removed from the tube and then soaked in a solution of SDS to allow its binding to the partially separated protein. 3. The tube is then attached to an SDS phase slab gel which is oriented perpendicular to the first plane of the separation within the tube and placed within an electric field to allow further separation. 4. Following separation, the proteins are visualized by adding staining agents like silver nitrate or by Comasi blue dye. In recent times, more sensitive fluorescent stains known as a SYPRO dye have been developed. These dyes do not introduce covalent modification into the protein. They have a broader linear range and are highly sensitive to the presence of protein. 5. In resulting gel after running and staining leads to the development of unique pattern of dots with each dot representing a single protein. Nucleic acid purification based on gel electrophoresis. The separation of nucleic acids such as a DNA is commonly carried out through the gel electrophoresis, although other procedures do not exist. But the simplicity of the gel electrophoresis method combined with its high resolution ability has been electrophoresis the most preferred method for DNA analysis. However, in contrast to protein, the gel matrix is composed of agarose rather than polyacrylamide. The agarose gel can separate DNA molecules having a size range of a few hundred base pair to about 20 kilobases. The negative charge present across DNA backbone arises from the phosphate group gives it a natural charge which is utilized in the electrophoresis where the DNA molecules moves away from the negative charge electrodes towards the positive charge electrodes. 1. The mechanism of separation of DNA molecules in an agarose gel electrophoresis is not particularly well understood. The migration of DNA molecules happens through the pore of gel matrix. The size of the pore depends upon the buffer composition in which the gel is prepared and concentration of agarose. 2. Originally, though to happen in snake bite like movement, repetition through the agarose matrix, which has later found to be by elastic motion with stretching in the direction of a positive charge followed by the contraction into a dense ball. The size of the ball being formed depend upon the pore size within the gel matrix formed through the cross link of agarose. 3. Pulse field gel ultraforces PFGE. The PFGE is more upgraded method for separation of DNA fragments having a size greater than 20 kilobases. In pulse field gel electrophorese, the orientation of the electric field web with respect to the gel is change at regular intervals to allow periodic alteration of the direction of DNA migration through the field. In this process, the direction, intensity, and the duration of the electric field is changed independent of each other. The difference between the direction of migration induced by each of the electric field is the reorientation angle and and the correspondent of the angle that the DNA must turn as it changes its direction of migration each time the field are switched. 4. The visualization of DNA in an agarose gel is done by adding the stain ethidium bromide ETBr, which intercalates within the major grooves of the DNA double helixes. The ethidium bromide binding results in visible fluorescence when the gel is observed under the UV light. However, the major disadvantage in using the ethidium bromide is that the reagent is highly carcinogenic and thus extreme care must be taken while handling it. Southern blotting. The different experimental techniques requires the nucleic acid labeling and hybridization, which are mostly used for identification and quantification of specific nucleic acid sequences. The procedure of blotting involves the immobilization of a sample DNA onto a solid matrix, which is generally nylon or of nitrocellulose in nature. The procedure of DNA blotting was first developed by Edward Southern, after whose name the process has been named. 1. Southern blotting involves the agarose gel after electrophoresis to be placed on stack of filter paper ends of which remains dipped in a reservoir containing a buffer solution that helps in transfer. 2. The hybridization membrane or the nitrocellulose membrane is placed between the agarose gel and another stack of paper towel that draws the transfer buffer upwards by capillary action and transfers the DNA from the agarose gel onto hybridization membrane. 3. For efficient transfer of DNA, the agarose gel is pre-treated in a depuration treatment with HCl 0.25 molar per liter, which is followed by the treatment with alkali. This procedure shortens the DNA fragments by alkaline hydrolysis at depurination site. This also leads to denaturation of double-stranded DNA into a single-stranded DNA, making it accessible for probing. 4. The following transfer DNA is fixed to the membrane by over-baking the membrane at 80 degrees centigrade. Due to the inflammable nature of nitrocellulose, this process is done in the absence of air in a vacuum. 5. After fixation, the membrane is then transferred into a solution containing the probe designed for identification and quantification of targeted isolated DNA sequences. Northern blotting. The blotting procedure involving transfer of RNA is termed as northern blotting, which is an extension of southern blotting procedure and was developed by James Alvin, David Kemp, and George Stark. Initially, RNA was found not to bind with nitrocellulose, hence the transfer membrane used was benzyloxymethyl DBM paper, which is prepared by diazotization of amino benzyl oxymethyl paper, which in turn can prepare from Wortmann 540 paper. The RNA bind covalently with the diazobenzyl oxymethyl paper, which is then available for hybridization reactions. The hybridization RNA can be visualized by autoradiography, which is also a viable technique for southern blotting. In subsequent years, the methodology of binding RNA to nitrocellulose membrane was established. Western blotting, the procedure for transferring electrophorous protein from a polyacrylamide gel to a nitrocellulose or nylon transfer membrane, is termed as a Western blotting and was designed by W. Neil Burnetti. The bound protein are then available for further analysis by a variety of specific protein ligand interactions. Of the different methods commonly employed for detection of protein, the antibody are used for the detection of antigens. The lectins are often used to identify glycoproteins. In some cases, the tagged design probes are used to bind to specific protein, which are later be visualized by the fluorescence or autoradiography. The molecular cloning of DNA and RNA. DNA cloning. The term clone stands for an identical copy of original. The DNA cloning involves isolation specific gene or a stretch of DNA from a large counterpart followed by attaching to a small molecules of carrier DNA, followed by multiple replication of this modified DNA up to 1,000 folds by repeated replication of host cell containing it, or by creating multiple copies of modified DNA within the host cell. The cloning of DNA thus comprises of 5 important steps. 1. Cutting of target DNA at specific sites by using the enzyme called restriction endonuclease that cuts the DNA at specific sequences. 2. Cloning vectors or small molecules of DNA that are capable of self-replicating. These are most commonly plasmid DNA or viral DNA. 3. Using the DNA ligase to join the clone vector with the already cut target DNA, leading to the formation of a recombinant DNA. 4. The recombinant DNA to a host cell that provides the enzymatic machinery requires for duplicating recombinant DNA multiple types. 5. Identifying the host cells that has a successful taken the recombinant DNA. This procedure comprises what can be termed as recombinant DNA technology, and the most common host or system utilized for processing involves the bacteria E. coli, and its ideal system, due to its DNA metabolism, is well understood. A host of different vectors are associated with E. coli, and the technique for introducing recombinant DNA into a host cell host will develop in case of E. coli. Restriction endonuclease. The restriction endonuclease are enzymes that recognizes specific DNA sequences and introduces cuts in the double stranded DNA helix at that site, which leads to generation of specific fragments. The restriction endonuclease were discovered by Werner Erber in 1960 and are found across a wide range of bacterial species. The restriction endonuclease acts as a protection against the viral infection that introduce foreign DNA in the bacterial cell. The host DNA is protected from the digestion activity of enzymes by methylation of the host DNA, catalyzed by a specific DNA methylase, which is often paired with the specific restriction endonucleases, forming a restriction modification system. Generally, there are 4 types of restriction endonuclease present across the bacterial system. Type of restriction endonuclease. Type 1. The type 1 comprises of different subunits that are associated with the recognition, methylates a single site and cleaves DNA at a site of 1,000 base pair away from the site of methylation, requires the ATP for reactions. Type 2. The type 2 comprises of 2 different enzymes which are symmetrical and participate in either cleaving or modifying DNA sequences. The cleavage occurs within the recognition site itself, and reaction does not require any ATP. Type 3. The type 3 comprises of 2 different subunits, one of which recognizes the binding site and modifies the DNA, while the other function in cleavage. The recognize and methylates the same site while introducing a cut between 24 to 26 base pair away, requires ATP for reactions. Type 2s. Two two different enzymes, but recognition sequence is asymmetric. The cleavage occurs on one side of the recognition sequences up to 20 base pair away. The thousands of restriction internucleases are known to exist in bacterial system, and over 100 different DNA sequences have been identified that can act as a binding sites for more than one restriction enzymes. These recognition sequences are usually 4 to 6 base pair long and a palindromic in nature, that is read same from any direction, whether reading right to left or left to right. The restriction internuclease can introduce cut, which generates two types of ends in the DNA chains. Sticky and certain restriction internuclease makes the staggered cuts that leaves 2 to 4 unpaired bases on each end of the cut. These unpaired ends are termed as sticky ends, and they can base pair with the similarly cut complementary unpaired ends. Blunt ends. Other restriction internuclease cleaves both strands of DNA at the opposing phosphodiester bonds, leaving no unpaired bases on the ends, forming the so-called blunt ends. The size and frequency of restriction site. Since the restriction products depends upon the frequency of restriction sites, which depends upon the size of the restriction site. Now in a DNA molecules there can be 4 different nitrogen bases that accounts for DNA sequences. A sequences having a length of 6 base pair thus has probability of being present once every 4 to the power of 6 is equal to 4096 base pair. Similarly, if the length of the restriction site is 4 base pair, the probability frequency of the restriction site is once in 4 to the power of 4 is equal to 256 base pair. However, in a normal genomic DNA, the frequency is relative, is far less than its probabilistic value, as all the 4 nucleotides are not equally present. The following restriction digestion, the digestion products are separated by agarose gel or by HPLC to known site. After the target DNA fragment is isolated, the DNA ligases can use to join it to a similarly digested clone vectors, that is a vector digested by the same restriction endonuclease. Therefore, a target sequence digested with the enzymes, like for example, E. coli R1 can only be ligated with the vector DNAs that have been digested by the same enzymes and cannot be ligated with the vector cut with the different restriction enzymes. 1. DNA ligase. The DNA ligase catalyzes the phosphodiester bond formation between the restriction product and the vector DNA and requires ATP for completion of the reactions. 2. The sticky ends facilitates the bond formation more easily in comparison to blunt ends. However, the blunt ends can be modified to form sticky ends by the action of a bacteriophage lambda and the terminal transferase that add complementary homopolymeric tails. Nomenclature of restriction enzymes. An uniform method of nomenclature of restriction enzymes was proposed by Smith and Nathans in 1973. The key points in this nomenclature are: 1. The species name of the host organism is identified by the first letter of the genus name and the first 2 letters of the specific epithet to generate a 3-letter abbreviation. This abbreviation is always written in italic. 2. Where a particular strain have been the source, then this is identified by writing the strain name in non-italicized letter in capital letter. 3. If a particular strain code for multiple restriction enzymes, then the order of the discovery of each enzyme is presented in Roman numerals. Thus, in the name of E. coli R1, the first 3 italic letter refers to its source organism, E. coli, SHA coli, having strain or capital R, and it is the first restriction enzyme isolated from this strain. Hence, the Roman numerals 1. Transformation. The process of transformation involves the incubation host cell, E. coli cells, and the recombinant DNA together at a temperature of 0 degrees centigrade in a solution of calcium chloride. The mixture is then given a thermal shock by evaluating elevating the temperature to about 37 degrees centigrade to 43 degrees centigrade. This process makes the cell more permeable to foreign recombinant DNA, which then enters the cell. The electroporation is an alternative process that utilizes the incubation mixture to pulse of high voltage electric shocks. This also increases the permeability of host cells and allows the uptake of recombinant DNA. Quality check. However, despite these processes, the frequency of successful uptake of recombinant DNA is extremely low. Therefore, the method exists to identify those cells that have successful uptaken the DNA. This process involves the presence of certain antibiotic resistance genes that renders the host cell containing it to become resistant to the presence of that specific antibiotic. This process is used to specifically select those cells that contains the recombinant DNA and are selectively grown in increase the number of clones. Blue-white screening. The blue-white screening is a method of assessing the quality of a cloning procedure. In this procedure, the plasmid used for cloning must have the LAC-Z gene sequence within which exists the identification site. Quality check. However, despite those processes, the frequency of successful uptake of recombinant DNA is extremely low. Therefore, the method exists to identify those cells that have successful upon taken the DNA. This process involves the presence of certain antibiotic resistance genes that renders the host cell containing it to become resistant to the presence of that specific antibiotic. This process is used to specifically select those cells that contains the recombinant DNA and are selectively grown to increase the number of clones. Blue-white screening. The blue-white screening is a method of assessing the quality of cloning procedure. In this process, the plasmid used for clone must have the LAC-Z gene sequence within which exists the identification site of a or more multiple restriction digestion site. Therefore, a successful restriction of the foreign DNA will leads to the disruption of LAC-Z gene. The transformants are plated on the media containing the beta galactosidase substrate X gene. If the LAC-Z gene remains intact after digestion and ligation, it will give rise to a blue colony. If any degradation of the cut end occurs, then a white colony will be produced. Then upon plating clones, white colony formed within the plate or the cells containing successful transformed host cells contain the cloned DNA fragments. Culture maintenance. Following the identification of cells successfully transformed with recombinant DNA, the successfully cloned DNA is maintained within its host cell in liquid culture. Longer duration of storage requires the cell to be stored in extremely low temperature, which can be thawed later upon requirement. Expression of cloned genes. The cloning of genes of interest mainly stems from an interest in the product encoded by these genes. With the improvements happening in the understanding of DNA, RNA, and protein metabolism, the vector system have been developed to not only clone eukaryotic genes within an E. coli, but also to produce the end product of that target gene. The vector engineered to expresses eukaryotic protein by using the bacterial translational machinery are called expression vector. These vectors are also used to code for RNase probe from the cloned gene product and to purify large amount of the gene product. 1. The rate of expression of a cloned gene is controlled by replacing the gene own promoter and regulator sequences with more efficient that are engineered into these expression vectors. 2. A promoter and regulator sequence are usually placed near several unique restriction sites that serves as a cloning site for target DNA to allow the transcription of the insertion gene by RNA polymerase. These promoters have been modified to successfully bind E. coli RNA polymerase. 3. The E. coli RNA polymerase is a multi-subunit enzyme with its core being composed of two identical alpha subunit and one each of a beta and beta subunit. The activation of a core polymerase enzyme requires the binding of sigma factors, which initially binds the promoter region and then allows the binding of RNA polymerase to the promoter. 4. The most common promoter used in expression vector binds this sigma 70 factor. One of the most common such promoter is located 35 base pair upstream from the transcription initiation point 5 prime TTGACA 3 prime and the 10 base pair upstream, and it is known as a Pribnox box 5 prime TATA 3 prime. Both sequences must be bound by RNA polymerases in order to initiate the transcription. 5. Upstream regulators of the inserted DNA are usually A plus T rich and increases the rate of transcription by binding with the alpha subunit of the RNA polymerase. 6. Upon initiation of transcription by RNA polymerase, the process continues until encountering a transcription termination site along the stretch. The transcription ends with an invert sequence followed by a stretch of A residue. The most expression vector used for factor dependent transmission of transcription termination term, the most expression vector used for factor dependent termination of transcription expression in bacterial cell. The gene expressed in bacteria involves the transcription of mRNA from the incorporated foreign gene and translation of that mRNA into protein by a bacterial protein synthesis machinery. These two processes are closely linked in bacteria. The transcription is initiated by RNA polymerase binding to the promoter region of the gene. As the 5' end of the transcript is made, the ribosome immediately attached to the nascent RNA and begins to process of translation. Thus, the protein synthesis is initiated simultaneously as mRNA synthesis is happening. The entire process can be completed within a few minutes. One major consideration in expressing a foreign gene in bacteria is that the fact that the expressed product may be toxic to host bacterial cell. Allowing the gene to constantly transcribe and translate could ultimately result in the death of the host cells. Sometimes, even if the protein is not toxic, large quantities of the product can lead to the formation of exclusion bodies containing aggregates of insoluble protein. This leads to difficulty in recovering the product. The purification of expressed protein may be carried out by using the affinity tags. These are the DNA sequences that are fused to the coding region of the gene on its 5' end or its 3' end. These sequences are translated alongside the target protein in the form of fusion protein. This allows easy purification of target protein by using of affinity chromatography. Expression of a gene cloned genes in plant cells. Quality check. However, despite these processes, the frequency of successful uptake of recombinant DNA is extremely low. Therefore, the method exists to identify those cells that have successfully been taken the DNA. This process involves the presence of certain antibiotic resistant genes that renders the host cell containing it to become resistant to the presence of that specific antibiotic. This process is used to specifically select those cells that contains the recombinant DNA and are selectively grown to increase the number of clones. Blue-white screening. The blue-white screening is a method of assessing the quality of the cloning procedure. In this process, the plasmid used for cloning must have a LAGZ gene sequence within which exists the identification site of a or multiple restriction digestion site. Therefore, a successful insertion of a foreign DNA which leads to the disruption of LAGZ gene. The transformants are plated on the media containing beta-galactosidase substrate XGAL. If the LAGZ gene remains intact after the digestion and ligation, it will give rise to a blue colony. If any degradation of the cut end occurs, then a white color will be produced. Thus, upon plating colony, the white color formed within the plates or the cell containing successful transformed host cell containing in the cloned DNA fragment. Culture maintenance. The following identification of the cell successfully transformed with the recombinant DNA. The successful clone DNA is maintained within its host cell in liquid culture. Long duration of the shortage requires the cell to be stored in extremely low temperature, which can be thawed later upon requirement. Expression of cloned genes. The cloning of gene of interest mainly stems from an interest in the product encoded by these genes. With improvements happening in the understanding of DNA, RNA, and protein metabolism, vector system have been developed to not only clone eukaryotic genes within an E. coli cells, but also to produce the end product of that target genes. Vectors engineered to express eukaryotic proteins by using bacterial translational machinery are called expression vectors. These vectors are also used to code for RNase probe from the cloned gene products and to purify large amount of gene product. 1. The rate of expression of a cloned gene is controlled by replacing the gene own promoter and regulator sequences with more efficient that are engineered into these expression vectors. 2. A promoter and regulator sequence are usually placed near several unique restriction sites that serves as a cloning site for target DNA to allow transcription of the insert gene by RNA polymerase. These promoters have been modified to successfully bind E. coli RNA polymerases. 3. The E. coli RNA polymerase is a multi-subunit enzyme, which its core being composed of two identical alpha subunit and one each of beta and beta dash subunit. The activation of a core polymerase enzyme requires the binding of sigma factors, which initially binds to the promoter region and then allows the binding of RNA polymerase to the promoter. 4. The most common promoter used in expression vector binds to sigma 70 factor. One of the most common such promoter is located 35 base pair upstream from the transcription initiation point 5' TTGACA 3' and 10 base pair upstream, and it is known as a prime now box 5' TATA 3'. Both sequences must be bound by RNA polymerases in order to initiate the transcription. 5. The upstream regulators of the inserted DNA are usually A plus T rich and increases the rate of transcription by binding with the alpha subunit of the RNA polymerases. 6. Upon initiation of transcription by RNA polymerase, the process continues until encountering a transcription termination site along the stretch. The transcription ends with an inverted sequences followed by a stretch of A residue. The most expression vector usually a factor-dependent termination of transcription. Expression in bacterial cells. The gene expression in bacteria involves the transcription of mRNA from incorporating foreign gene and a translation of that mRNA into the protein by the bacterial protein synthesis machinery. These two processes are closely linked in bacteria. The transcription is initiated by RNA polymerase binding to the promoter region of the gene. As the 5' end of the transcript is made, the ribosome immediately attached to the nascent mRNA and begins the process of translation. Thus, the protein synthesis is initiated simultaneously as mRNA synthesis is happening. The entire process can be completed within a few minutes. One major consideration in expressing a foreign gene in bacteria is that the fact that the expressed product may be toxic to the host bacterial cell. Allowing the gene to constantly transcribe and translate could ultimately result in the death of the host cell. Sometimes, even if the protein is not toxic, the large quantities of the product can lead to the formation of inclusion bodies containing aggregates of insoluble protein. This leads to difficulty in recovering the product. The purification of a expressed protein may be carried out by using the affinity bags. These are the DNA sequences that are fused to the coding region of the gene on its 5' end or its 3' end. These sequences are translated alongside the target protein in the form of a fusion protein. This allows easy purification of the target protein by using the affinity chromatography. Expression of a expression of cloned gene in plant cells. The introduction of recombinant DNA into a plant has enormous potential for agriculture, producing more nutrients and higher yield crop that are resistant to the environmental stress such as insect pests, disease, cold, and drought. Unlike animals, the fertile plants of some species may be generated from a single transformed cells. Thus, a gene introduced into a plant cell may ultimately be transmitted to progeny through seed in successive generation. As with all system, the cloning in plant cells has its own peculiar problems. No naturally occurring plasmids have been found in plants to facilitate these processes, and thus the most challenging task is getting DNA into the plant cells. 1. Circumnavigation of this problem has been achieved through the soil bacterium Agrobacterium tumefaciens, which contains a large, approximately 2 lakhs base pair plasmid called the TL plasmid. 2. When the bacterium contacts a plant cell, a segment of plasmid called a tDNA, approximately 23,000 base pairs, is transferred from the TA plasmid to the plant cell's nucleus and is integrated at the random position in one of the plant's chromosomes during transformation. 3. The transfer of tDNA from Agrobacterium to the plant cell nucleus is mediated by 225 base pair repeats that flank the tDNA and by the products of several genes called virulence genes and located elsewhere on the TA plasmid. 4. The gene in the virulence regions are grouped into the operon virulence A, B, C, D, E, F, G, which codes for the enzymes responsible for mediating conjugative transfer of tDNA to plant cells. 1. Virulent A codes for receptors which reacts into the presence of phenolic compounds such as syringaldehyde or acetovanillones, which leaks out of damaged plant tissue. 2. The virulent B encode proteins which produces a pore or pilus-like structure. 3. The virulent C binds the overdrive sequences. 4. The virulence D1 and virulence D2 produces endonucleases which targets the target repeat border of tDNA segments. The VIR D4 is the coupling protein. 5. The VIR-E binds to the T strand protecting it from the nucleus attack and intercalate with lipid to form channels in the plant membranes through which the T complex passes. Begins with the right border. 6. The VIRG activates a VIR gene expression after binding to a consensus sequence once it has been phosphorylated by the VIRA. 5. The bacterial system that transforms tDNA into the plant genome can be utilized to transfer recombinant DNA. The Agrobacterium TA plasmid has been engineered in 2 ways. First is the TA plasmid from which the tDNA segment has been removed. The second is an Agrobacterium, the E. coli shuttle vector that contains the tDNA 25 base pair repeats flanks the gene that researchers want to introduce and a suitable marker, example kanamycin. 6. The engineered agrobacterium is used to infect the plant cells which then grow to form a transgenic plants. The virulent gene the products that are altered TA plasmid directs the transformation of the plant cell by the gene flanked by the tDNA 25 base pair repeats in the second plasmid. The transformed cell can be selected by growth on kanamycin and induces with the growth hormones to form new plants. 7. The entire process is initiated by activation of VIR-A, VIR-G complexes, which is followed by the production of single-stranded tDNA by VIR-D1 and VIR-D2 proteins. This leads to the activation of VIR-B and VIR-D4, the type 4 secretion. Then the VAR E2 interacts with the VIP1 and VAR E3 VAR3 to allow integration. Agrobacterium tumefaciens, expression of cloned genes in animal cells. There are several common methods for introducing DNA into the animal cells. Because no suitable plasmids like vectors is available, the transformation requires the integration of DNA into the host cell chromosomes. One, spontaneous uptake is a procedure in which the calcium phosphate DNA precipitate is taken up by the cells through endocytosis. Generally, only about 1 in 10 to the power of 2 to 10 to the power of 4 cells is transformed in this procedure. 2. Another method to make the cell transiently permeable to DNA by exposing them to a brief high voltage pulse in the technique called electroporation. 3. Microinjection. The microinjection is a procedure that involves direct injection of DNA into the nucleus of cell with the aid of a very fine needle. However, as cells must be injected one by one, the total number that can be treated is small. 4. The eukaryotic viruses are utilized to introduce foreign DNA into the chromosome in a host cell. Certain retroviruses have been modified to act as viral vectors to introduce foreign DNA into the mammalian cells. The viruses have their own mechanism for integrating the foreign DNA into a cell to form transgenic animals. When the virus enters the cells, its RNA genome is converted into DNA by reverse transcriptase and then integrated into the host genome in a reaction mediated by the viral integrase. Genomic library. The genomic library refers to many recombinant DNA that together contains a complete collection of all the DNA sequences in the entire genome of an organism. The construction of genomic library is of importance as it acts as a detailed repository of instruction contained within an organism's genome. The step of forming a genomic library can be summarized as 1. The digestion of total genomic DNA with restriction endonucleases which results in fragments ranging in all sizes with the size that are compatible for insertion within the cloning vector containing virtually all sequences are represented among the clones. Smaller size fragments are removed from the mixture of centrifugation or electroporation. 2. The insertion of the digestion product within the suitable vector, also digested by the same restriction enzyme, followed by the ligation of the fragments to be cloned. 3. The ligated DNA mixture is then used to transform bacteria or yeast cell to produce a library. 4. Using hybridization method, the clones having overlap in sequences can be identified as a set of overlapping clones represents a catalog of a long continuous segment of a genome referred to as a contig. 5. The segments that have been already mapped on the chromosome can be used to determine its location within a genome. A well-organized library is usually composed of thousands of long contigs, all of which are arranged orderly on a detailed physical map. 6. The sequence tagged sites (STS) or such known sequences within the library that have their location known on the physical map and can provide landmark for genomic sequence projects. See chromosomal DNA library. A chromosomal DNA library is composed of sequences that are only expressed to transcribe mRNA from them. These library are of importance as more whole genome sequences get produced. The chromosomal DNA library lacks the non-coding part of the genome that makes up a large proportions of the eukaryotic genome. The chromosomal DNA library are formed by 1. The mRNA from an organism or from a group of specified cells of an organism is first extracted. 2. From the mRNA, the double-stranded DNA is produced by the multi-step reactions involving the enzyme reverse transcriptase. 3. The resulting double-stranded DNA fragments are then inserted into a suitable vector and cloned with which creates a population of clones. 4. The search for a particular gene is carried out by focusing on a chromosomal DNA library generated from the mRNA of a cell known to express the gene. 5. To aid in the mapping of large genome, the chromosomal DNA in a library can be primarily sequenced at random to produce a useful type of STS called as a expression sequence tag EST. The expressed sequence tag, ranging in size from a few dozen to several hundreds base pair, can be positioned within the large genome map, providing marker for expressed gene. A chromosomal DNA library can be made more specific by cloning a chromosomal DNA or chromosomal DNA fragments into a vector which has a reporter gene fused on the chromosomal DNA insert. This is termed as a reporter construct. The two useful markers utilized for producing a reporter construct are the green fluorescent protein GFT and a epitope tags. The one expression of green fluorescent protein tagged with the protein of interest generates a fluorescent protein which can be visualized by its fluorescence, which can be observed under microscope to know the location of the protein within a cell. 2. An epitope tag is a short protein sequence that can be bound by a well characterized antibody. The tagged protein can be specifically precipitated from a crude protein extract by insertion with the antibody. If any other protein binds to the tagged protein, those will precipitate as well, providing information about the protein-protein interaction in a cell. The vector used for clone library preparation. Plasmid. The plasmid are small circular DNA molecules that naturally occurs within the microbial cell but are not part of microbial chromosomes. The plasmid ranges in size from 5,000 to 40,000 base pairs. They can be introduced into the bacterial cells by a process of transformation or by electroporation, as discussed before. The transformation of bacterial cell with purified DNA, never a very efficient process, becomes less successful as plasmid size increases, and it is difficult to clone the DNA segment longer than about 15,000 base pair when plasmid are used as vector. The investigator have developed a host of different plasmid aimed for clone library preparation, the most widely used for which is the vector PBR322. PBR322. E. coli plasmid PBR322 is a artificially designed plasmid vector that has all the characteristics of an ideal plasmid vectors and is widely used. The characteristics that makes PBR322 more important are: 1. PBR322 has a site for origin of replication. The site acts as a binding site of enzyme that facilitates DNA replication at a higher rate, generating 10 to 20 copies per cell. 2. The plasmid containing two antibiotic resistant genes for tetracycline resistant and ampicillin resistant, which allows identification of cell containing PBR322. 3. The PBR322 has multiple sites for different restriction internuclease. Example: PST1, BAMH1, E. coli R1, SAL1, PVU2. These sites acts as a region where DNA can be cut and DNA to be cloned can be inserted. 4. The PBR2022 has very small size, 4,361 base pairs, that facilitates its easy entry into the host cells and allows greater transformation frequency. Bacteriophage. The formation of a recombinant DNA having a large size insert is not possible using plasmid vector. The bacteriophage lambda can be utilized as a vector and have a size of 48,503 base pair. The factor that gives this attribute to bacteriophage vectors are: 1. Approximately one-third of lambda genome is not essential and can be replaced with foreign DNA. 2. The DNA is transferred by packing into infectious phage particles that can carry 40,000 to 53,000 base pair. This makes sure that only recombinant DNA of appropriate size can only be transferred. The bacteriophage lambda has been designed by researchers to contain 3 regions, 2 of which contains essential genes for replication, while the third region is not essential and it is replaced by the target sequence to be cloned. The upper limit for inserted product is to be cloning vector is about 23,000 base pairs. The following successful ligation resulting recombination DNA is packed into phage particles, which is done by incubating replicating the recombinant DNA with bacterial cell extract that contains the protein required for forge particles formation. This step is formed by in vitro packing, which then transmits the recombinant DNA into E. coli cells. Plasmids. The plasmid are also known as a forgemid or artificial design cloned vectors that are designed by having both properties of bacteriophage and plasmid. This vector has origin sites from plasmid along with the presence of F1 origin of replication from F1 bacteriophage. The plasmid are used as a cloning vector in combination with the filamentous phage M13. The the phasmids are known as a phagemids or artificially designed cloning vectors that are characterized by having both the properties of bacteriophage and plasmids. This vector has a origin sites ORI from the plasmid along with the presence of F1 origin of replication from F1 bacteriophage. The phasmid are used as a cloning vector in combination with the filamentous phage M13. A phasmid can be replicated as a plasmid and can be packed as a single stranded DNA in viral particles. The phasmid contains a origin of replication ORI for double stranded replication as well as F1 origin to enable the single stranded replication and packing into the phage particles. The use of the M13 in combination with the phasmid has a definite advantages over bacteriophage lambda, as it does not causes lysis in bacterial cell but forms plaques by slowing down the growth of bacterial cell. Cosmid. The cosmid are cloning vectors that are engineered from plasmid by introducing a specific site from the bacteriophage's lambda known as a cos site. The cos site is an abbreviation for the cohesive end site. The cos site or specifically speciality the cos site or a speciality of the lambda phage, which can be linearized to fit into the phage head but circularized into the host cells. To achieve this, it has the cohesive or sticky site at each end. These cosmids can be used as a gene cloning vector in conjunction with the in vitro packing system observed in the bacteriophage. The cosmid vectors having a site of 5 kb can carry an insert of 32 to 47 kb in the size of foreign DNA. The recombinant cosmid DNA is injected and circularized like phage DNA but replicates as a normal plasmid without the expression of any phage functions. The transformed cells are selected based on the vector drug resistant markers. Engineered cosmids like a PWE SCOs are characterized by the presence of multiple cloning sites for cloning using non-size selected DNA. 2. The phage promoters planking the cloning site. 3. Unique sites for rate cutter such as NOT 1, NSAC2 and SFI1. Bacterial artificial chromosome BAC. The bacterial artificial chromosome was developed by Shizuya et al. In 1992 and it is based upon the single copy sex factor F of E. coli. The bacterial artificial chromosome contains a lambda cos n site and P1 logs P site along with the 2 cloning sites for digestion by HIND3 and HAM H1 and several restriction enzyme sites, example SFI1 and NOT1. The site of insertion of the product to be cloned is flanked by T7 and SP6 promoter region, which are used to generate RNA probe. The bacterial artificial chromosome can be introduced into the host cell without requiring any packaging and it is very efficient. The bacterial artificial chromosome can carry insert up to 300 kilobase in size and can be maintain them for 100 generations. 1. The 2 most common bacterial artificial chromosome used are P below BAC11 and PEC BAC1, which contains LAC-Z gene carrying a multiple cloning sites. 2. The P below BAC11 has has two equally R1 sites, eco R1 sites, one of which is located within the lag Z gene, while another is located within a CMR gene stretch. 3. The improvement on this has been made by replacing the lag Z and SAC-B gene. The insertional inactivation, that is inactivation of the gene by insert of a DNA stretch of SAC-B, results in growth and of host cells in sucrose containing medium. Yeast artificial chromosomes. The requirement associated with the large size genome and high capacity cloning vector have led to the development of yeast artificial chromosome YACs. The yeast artificial chromosome vector can carry significantly large amount of DNA sequences originating from eukaryote and are usually grown within the nucleus of yeast cell, in contrast to standard E. coli based culture method. The yeast artificial chromosome vector are characterized by the presence of yeast origin of replication. The two selectable markers and specified sequence derived from telomeric and centromeric regions of eukaryotic DNA that aid in their proper segregation during cell division and also in protection. 1. Prior to cloning, the yeast artificial chromosome present in a circular form similar to plasmid, which is then cleaved with a restriction endonuclease. This digestion is usually carried out using the BAM-H1 and removes the region between the two telomeric sequences. 2. Following this, another digestion is performed by ECO-R1 within an internal site, which divides the vector into two parts known as a vector arm. 3. Each having a different selectable markers. Yeast host cells. Apart from E. coli cells, the yeast cells are particularly well suited for cloning of eukaryotic genes. The most commonly used yeast cells is Saccharomyces cerevisiae, having a size of 14 to 10 to the power of 6 base pair, and the entire sequence is known along with the detailed understanding of these processes. The yeast cells are very easy to maintain and can be grown on large scale. The plasmid vectors have also been designed to be cloned within the yeast cells. Some recombinant plasmid incorporate multiple replication origin and other elements that allows them to use in more than one copies. For example, yeast or E. coli, the plasmids that can be propagated in cells of two or more different species are called shuttle vectors. Isolation of specific DNA sequences. The DNA hybridization is the most sequence based processes for detecting a gene or stretch of sequences. The basic processes of this method involves radio labeling probes composed of DNA or RNA that are complementary to the sequences being sought. This classical approach is used to detect a DNA sequences within a collection of DNA clones known as a library. 1. In a library on agar plate culture containing multiple colonies of host bacterial cell growing, with each colony having a different recombinant DNA. 2. The nitrocellulose paper is pressed onto an agar plate containing many individual bacterial colonies from the library. 3. Some cells from each colony adheres to the paper, forming a replica of the plate. 4. The paper is treated with alkali to disrupt the cell and denatures the DNA within, which remains bound to the region of the paper around the colony from which it came. 5. The radioactive DNA probe designed to identify specific target gene are added, which then anneals only to its complementary DNA. 6. After any annealing probe DNA is washed away, the hybridized DNA can be detected by autoradiography. The autoradiography is a process of localizing and recording of a radio label within a solid phase, where an image is produced on the photographic emulsion formed of silver halide crystals suspended in a phase of gelatin. When a beta particles or gamma rays from the radionuclide pass through the emulsion, the silver ions are converted to silver atoms. This results in a latent image being produced, which is converted to a visible images. When the image is developed, the development is a system of amplification in which the silver atoms causes the entire silver halide crystals to be reduced to metallic silver. For hybridization detection, the sample is placed in intimate contact with the film, and radioactive emission from the radio labeled probe leads to the production of a black area on the autoradiograph. This procedure works best when the sample is radio labeled with weak to medium strength alpha gamma strength emitting alpha emitting radionuclides such as H3 carbon 14 and S35. In vitro mutagenesis and deletion. Site directed mutagenesis. The cloning technique are used not only to increases the production of any protein, but can also be used to produce proteins products that are slightly altered from their native forms. The site directed mutagenesis is a method where the specific amino acids may be replaced individually. This method is utilized to study the protein structure and functions by altering the cloned DNA sequences. Classically, mutant are generated by treating the target organisms with chemical and physical agents known as mutagens. However, this process is much inferior to site-directed mutagenesis, as the process is not specific and can affect any gene, thus giving no guarantee that the gene of interest has been successfully mutated. Primary extension. A single primer method involves in vitro DNA synthesis with a chemically synthesized oligonucleotide having a length of 7 to 20 nucleotide. The oligonucleotide carry a base mismatch of complementary sequences. The procedure requires the target DNA to be present in single stranded form. 2. The synthesis of oligonucleotide primer initiates DNA synthesis and is itself incorporated into the resulting heteroduplex DNA. This DNA is transformed into E. coli cells. 3. Within the host cells, the heteroduplex give rise to homoduplexes whose sequences are either that the original wild type DNA or that containing the mutated base. 3. The frequency of the mutated clone is much less than the clone containing wild type sequences. The mutants can be isolated by hybridization with the P32 labeled oligonucleotide or as probes. 4. The presence of mutations can best detected by sequencing the isolated clones and the vectors contained within. This method, however, has many deficiencies. The generated double-stranded heteroduplex molecules is contaminated both by a single-stranded non-mutated template DNA and by partially double-stranded molecules. This considerably reduces the number of mutant DNA. The combination can be removed by density gradient centrifugation. Gene knock and slicing in bacteria. The gene knockout and slicing in bacteria. Gene knockout and slicing is a method of studying the function of a product produced by a specific gene by preventing the formation of the product by introduction of deleterious mutations within a gene or by somehow disruption the mechanism of mRNA or protein synthesis. In bacterial cell, the gene splicing can be achieved through either complete removal of gene or by replacing with a mutated version. 1. The gene knockout in bacterial genome can be achieved by gene deletion through two sequential recombinations. 2. This is achieved by plasmids having homology sequences for regions on either side of the target gene, and it is inserted into the bacterial DNA by homologous recombination. 3. The second recombination reforms the plasmid. The procedure is chance based and can lead to two outcomes. Firstly, it can reproduce the original plasmid, while secondly, it can contains the target genes. Only is the second scenario the deletion will be successful. The various improvements and variations on above method have been used in years since. Additionally, in recent years, the completely different methods have been developed to silence gene without altering the DNA. One such example is to produce antisense RNA, AsRNAs, that binds the mRNA produced by the gene such that it cannot be translated, for example, as as antisense RNA to the ribosomal binding site, prevents mRNA or ribosome association, and simultaneously prevents the translation and promotes degradation. Gene knockout and slicing in eukaryotes. The analog of a system observed in bacteria are also used in eukaryotes to produce gene knockouts. One method that can permanently and hereditably mutate mammalian gene makes use of zinc finger nucleases. 1. Highly specific DNA binding domains from protein called zinc finger protein are conjugated to an endonuclease called FOK1 that only functions as a dimer. 2. When zinc fingers bind to a targeted sequence on both strands of a double-stranded DNA, the endonucleases are perfectly positioned to dimerize and form a double-stranded break. 3. When the cell repairs this, break it down so by a process called non-homologous end joining, which leads to insertion or deletion of DNA with relatively high frequency. 4. Gene targeting for slicing. As these errors or causes frame shifts, they destroy functions of gene products, allowing for its effect to be studied. Protein sequencing. One major methodological approach to ensure the proper integration of a mutation in a protein coding gene is to sequence the transcribed product and observe the changes in protein sequences. Studying the sequences of protein is also extremely important to identify the tertiary and quaternary structure and the binding properties and the protein's function. The first successful sequence of protein was carried out by Frederick Sanger, who sequenced the amino acid arrangement of insulin. The basic method developed by Sanger are still used to determine the sequence of amino acids. Sanger sequencing methodology. 1. Frederick Sanger developed the reagent 1-fluoro-2,4-dinitrobenzene FDNB, which can bind to amino terminus end of amino acid chain. Dancyl chloride and dapsyl chloride can also bind the amino terminal end, and the derivative is more easily detectable too. Following the amino terminal labeling, the amino acid chain is hydrolyzed to its constituent amino acid, and label 1 is detected. 3. The hydrolysis stage destroys the amino acid chain and cannot be used to sequence a polypeptide beyond its amino terminal residue. However, the process helps determines the number of chemically distinct polypeptides in a chain, provided each has a different amino terminal residue. 4. The sequencing of entire protein chain, a procedure developed by the Per Edman, is employed known as a Edman degradation. This procedure labels and removes only the amino terminal residue from a peptide while leaving the other peptide bound intact. 5. The peptide is reacted with the phenyl isothiocyanate under mildly alkaline condition, which converts the amino terminal amino acid to phenyl thiocarbamoyl PTC adduct. The peptide bond next to the phenyl thiocarbamoyl adduct is then cleaved in a step carried out in anhydrous trifluoroacetic acid with the removal of amino terminal amino acid as a anilino thiazolinone derivatives. 6. The amino acid derivative is extracted with organic solvents and it is converted to a more soluble, more stable form, phenyl thiohydantoin, through a treatment with aqueous acid, and then it is identified. 7. The use of a sequential reaction carried out under first basic then acidic condition provided control over the entire process. Each reactions with amino terminal amino acids can go essentially to completion without without affecting any of the other peptide bond in the peptide. 8. This procedure is sequentially repeated until the entire sequence is determined. The procedure of Edman degradation is carried out in a sequenator that mixes a reagent in required proportions, separates the product, identifies the derivatives, and result is recorded. Detection of post-translational modification in large proteins. The polypeptide, after synthesis from the translation, forms the primary protein structure. After this, the amino acid folds into itself based on disulfide bond formation between cis residue to form the secondary structure as a protein subunit. Such subunit can come together to form multi-subunit large size protein. The large size protein are sequenced by breaking down the protein into smaller size chain. First, the protein is cleaved into small chain by chemical or enzymatic method. 1. Breaking disulfide bond. The breaking disulfide bond is achieved by two methods. 1. By oxidation through performic acid, and 2. Through reduction by dithiotol. This step is important as a disulfide bond can interfere with the enzymatic and chemical degradation of the protein. 2. The polypeptide chain cleavage. This polypeptide chain cleavage can be attained by many ways. One common method is to digest through the protease, which hydrolyzes the peptide bond. Some proteases enzymes cleaves peptide bond adjacent to certain amino acids. The trypsin catalyzes hydrolysis of a peptide bond in which the carbonyl group is contributed by either a lysine or arginine residue, regardless of the length of amino acid chain. A polypeptide having 5 lysine or arginine residue will yield 6 small peptide chain if digested by trypsin. 3. The order of trypsin fragments in the original polypeptide chain must now be determined. Another sample of intact polypeptide is cleaved into fragments using a different enzymes or reagent. One of the cleavage peptide bonds at point other than those cleaved by trypsin. The polypeptide chain cleaves can be achieved by chymotrypsin, which cleaves at PHE trypsin tyrosine residue, while cyanogen bromide cleaves at methionine residue. 4. The sequence of each fragment is obtained by two cleavage procedures are examined with the objectives of finding peptide from the second procedure, whose sequence establishes the continuity by searching of overlaps. The overlapping peptide obtained from the second fragments yields the correct order of peptide fragments produced in the first. 5th, the location of a disulfide bond within a protein can be identified through an additional step followed sequencing. In this step, the sample of same protein is again cleaved by same enzymes as trypsin, but without breaking the disulfide bond. The resulting peptide are then separated by gel electrophoresis and are composed with the original peptide generated by trypsin digestion following disulfide bond breakage to identify the location of the disulfide bridge. DNA sequencing. The sequencing of DNA is another major pathway to study the genomes and changes within the different genomes across different organisms. The induced mutations and their incorporation can also be studied by observing the DNA sequences. The first method of DNA sequencing was developed by Wu and Taylor in 1977, which was used to sequence the cohesive end of the vector. A much more improved method was developed by Frederick Sanger in 1977. Maxam and Gilbert introduces an improved method of DNA sequencing in 1977. At the same time, Frederick Sanger developed his chain termination or dideoxy method. Among these two methods, the dideoxy method gradually becomes the most widely used method for obtaining DNA sequences. Maxim Gilbert sequencing. This method of DNA sequencing requires the reactive labeling of 52 end of the DNA fragments to be sequenced utilizing a kinase reaction using gamma p32 followed by purification of DNA. 1. The process involved generation of breaks at a small portion of one or two of the four nucleotide bases. 2. The purine A plus G was or depurinated using the formic acid followed by methylation of guanine residue and to some extent the adenine by dimethyl sulfate. 3. The pyrimidine C plus T are hydrolyzed using the hydrazine. The addition of salt sodium chloride to hydrazine inhibits the reaction happening to thiamine residue, thus restricting hydrolyzation of cytosine only. 4. The modified DNA may then be cleaved by using heated piperidine CH25NH at the site of the modified bases. The concentration of the modified chemicals is controlled to introduce on average one modification per DNA molecules. Thus, a series of labeled fragments is generated. 5. The fragments are in the four reactions are separated by running in denaturating acrylamide gel for size separation. The visualization of the fragment is carried out by autoradiography. The comparison based on presence and absence of certain fragments leads to the sequence base being inferred. Chain termination DNA sequencing. The chain terminator or D-deoxy procedure for DNA sequencing capitalized on two properties of DNA polymerases. 1. Ability to synthesize faithful complementary copy of a single standard DNA template. 2. Use of 2' 3' dideoxynucleotides as substrate for DNA chain elongation. 3. Incorporation of the modified substrate leads in termination of DNA chain elongation as dideoxynucleotides. Lacks a hydroxyl group at 32 end and cannot be used as a substrate for chain elongation. Thus, dideoxynucleotides acts as a chain terminator. 4. The major effect of DNA chain elongation is a chain termination method is the clean-out fragment of DNA polymerase as it lacks the 5' to 3' endonuclease activity, a property present in the intact DNA polymerase. 5. The DNA synthesis is carried out in the presence of the 4-deoxynucleoside triphosphate, one or more of which is labeled with phosphorus 32, and in 4 separate incubation mixes containing a low concentration of one each of the 4 dideoxynucleoside triphosphate analogues. Therefore, in each reaction, there is a population of particularly synthesized radioactive DNA molecules, each having a common 5' end, but each varying in length to a base specified 3' end. 6. After a suitable incubation period, the DNA in each mixture is denatured and electrophorosed in a sequencing gel. 7. A sequencing gel is a high-resolution gel is used to separate single-stranded DNA fragments based on their size and which are resolved fragments differing in length by a single base. These gels are composed of 6 to 20% polyacrylamide and urea. The function of the urea is to minimize DNA secondary structure, which affects the electrophoretic mobility. The gel is run at enough power to heat up at about 70 degrees centigrade, which minimizes the DNA secondary structure. The labeled DNA bands obtained after such electrophoresis are revealed by the autoradiography on the large sheet of X-ray films, and from these the sequence can be read. 8. The DNA to be sequenced is usually cloned into one of the clustered cloning sites in the LAC region of the M13 MP series of the vector. The cloning into this region is carried out by a large section of restriction enzymes but still permits the use of a single sequencing primer. 9. One of the variant of the classical approach often employed is in automated sequencing is a cycle sequencing. This involves a linear amplification of sequencing reaction using 25 cycles of renaturation annealing of the specific primer to one strand only, and the extension in the presence of tag DNA polymerase plus labeled dideoxynucleotide. Alternatively, labeled primer can be used with unlabeled dideoxynucleotides. 10. The direction of a generated stretch of DNA in automated sequencing processes do not employ an autoradiographic method, but rather use the fluorescence probe to facilitate easy detection. 11. In this procedure, the fluorescent tags are attached to the chain termination nucleotide. Each of the four dideoxynucleotide carries a spectrally different fluorophore. The tag is incorporated into the DNA molecules by the DNA polymerase and accomplishes two operation in one step. It terminates synthesis and it attaches the fluorophore to the end of the molecules. 11. By using four different fluorescent dyes, it is possible to ultrafluoresce all four chain terminating reactions together in one lane of the sequencing gel. The DNA band are detected by their fluorescence as the ultrafluoresce passed a detector. If the detector is made to scan horizontally across the base of the slab gel, many separate sequences can be scanned, one sequence per lane, because the different fluorophore affects the mobility of the fragments to different extent. The sophisticated software is incorporated into the scanning step to ensure that the band are read in the correct order. Polymerase chain reactions. A specified stretch of DNA can be amplified many folds of a polymerase chain reaction (PCR), a process central to different methods for investigating genetic sequence and genetic variations. The process was first developed by Carey Mullis in 1983, and since he became staple process any genetic investigations. The in PCR, if the sequence of flanking region of DNA is known, then the region lying in between can greatly amplified. The process of PCR is extremely simple in its basic form. 1. The two synthetic oligonucleotides are prepared complementary to the sequence on opposite strands of the target DNA at position just beyond the ends of the segment to be amplified. The oligonucleotide serves as a replication primer that can be extended by DNA polymerase. The 32 end of the hybridized probe are oriented towards each other and positioned to prime DNA synthesis across the desired DNA segment. 2. The PCR uses the heat-stable DNA polymerases such as Taq polymerase derived from the bacterium Thermus aquaticus that lives at a 90 degrees centigrade temperature of hot spring. The enzyme remains active after the heating step required for denaturation of double-stranded DNA to single-stranded forms to allow DNA synthesis. The thermostable nature of the enzyme remains. It does not have to replenish after each round of heating. 3. The 4-deoxynucleoside triphosphate are then added and the primer DNA segment is replicated selectively. The cycle is heating, cooling, replication is repeated 25 or 30 times over a few hours in an automated process, amplifying the DNA segment flanked by the primer until it can be readily analyzed or cloned. The PCR procedure has 3 steps. 1. Denaturation. The denaturation involves the breakdown of hydrogen bond between the two strands of double-stranded DNA to expose the single strand. The denaturation occurs usually at the temperature of 94 to 95 degrees centigrade. 2. Annealing is a process where the temperature is lowered to 50 to 56 degrees centigrade to allow the binding of a synthetic oligonucleotide primer to their complementary sequences, flanking the region to be amplified. 3. Extension is the last step of the process where Taq polymerase extends the DNA chain from the primer at a temperature of 72 degrees centigrade. These steps are cyclically repeated for 25 to 30 times to increase the copy number of the target DNA. The DNA amplified by the PCR can be cloned. The primary can includes non-complementary and that has a site for cleavage by the restriction endonuclease. Although these parts of the primer do not anneal to the target DNA, the PCR processes incorporates them into the DNA that is amplified. The cleavage of amplified fragments at this site creates sticky ends used in negation of amplification DNA to a cloning vector. Genome sequencing. An organism's genome is the entire genomic information presented within itself. One of the vast undertaking in understanding genomic data was undertaken in the 1980s in the form of International Human Genome Project. The completed genome was published in April 2003. The construction of whole genome sequences requires the generation of high density map. The high density map are of importance as the largest stretch of DNA that can be sequenced by the Sanger method in a single pass is 600 to 800 nucleotide, and many of the genome to be sequenced are over 1 million times greater. Therefore, it is necessary to break each genome into much smaller pieces of sequencing and then joining the pieces together again. However, joining the pieces together requires a marker because two fragments can only be shown to overlap if they have unique sequences, that is, no repeated sequences in common. For most organisms of interest, the density of genetic marker is woefully inadequate, and this has related to development of physical markers. RPLF, random fragment length polymorphism. The first physical map of genomes was prepared by random fragment length polymorphism RFLP markers. The RFLP markers was based on sequence polymorphism, that is single base pair changes that happen between different individuals in a frequency of 1 in every 1000 base pair. Some of the sequence changes affects the recognition site for the restriction enzymes, resulting in variation in the size of DNA fragments produced by digestion with the restriction enzymes. These variations or restriction fragment length polymorphisms. Bordstein et al. 1980 was the first to recognize that DNA probe could be used to identify the polymorphic sequences and to develop the RFLP as a marker for genomic map preparation. Such RFLP can be generated in certain ways. 1. Base change to facilitate the loss of restriction endonuclease site. 2. Insertion or deletion of stretch of DNA. 3. By having variation in number of repeats in microsatellite DNA. The probes used to generate RFLP map must target highly polymorphic sites. The first RF restriction fragment length polymorphism map of entire genome was developed for human genome. 1. Direction of RFLP was undertaken by hybridization to southern blot to DNA from 1680 clones from a forge library of 5 unrelated individuals. 2. The DNA from any one was digested with 6 to 9 restriction enzymes. Over 500 probes were identified that detected variable banding patterns indicates of polymorphism. 3. From this collection, a subset of 180 probes detecting the highest degree of polymorphism was selected for inheritance study in 21 3-generation human families. 4. The additional probes were generated from chromosomal specific library such that ultimately 393 restriction fragment length polymorphs were selected. 5. The various loci were arranged into linkage group represents the 23 human chromosomes by combination of mathematical linkages analysis and physical location of selected clones. The latter was achieved by hybridizing probe to panel of rodent human hybrid cells containing varying human chromosomal complements. Apart from genome mapping, RFLP is used in forensic as a tool of DNA fingerprinting. The process of DNA fingerprinting utilizes a RFLP is based on the following step. 1. The DNA fragments from digestion of genomic DNA by restriction endonuclease are separated by size electrophoresis. The double-stranded DNA is denatured by soaking the agarose gel in alkali and then blotted onto a nylon membrane to reproduce the distribution of fragments in the gel. 2. The member is immersed in a solution containing a radioactive labeled DNA probe. The probe for a sequence that is repeated several times in human genome generally identifies a few of thousands of DNA fragments generated when the human genome is digested with restriction endonucleases. 3. The autoradiography reveals the fragments to which the probe hybridization and probe RFLP pattern characteristics of individual whose GDNA is used. Genomic DNA is used. These RFLP pattern are inherited from parent to child and are widely used as a evidence for paternity testing and is a key tool of modern day forensic. Randomly amplified polymorphic DNA (RAPD). The polymorphic DNA can also be detected through the amplification of genomic DNA with a single primer of arbitrary nucleotide sequence. The primers were designed to be about 9 to 10 nucleotide in length, having 50 to 80% of G plus C content, and are devoid of palindromes. Not all the DNA amplified using these primer are always polymorphic, but the ones that are termed as randomly amplified polymorphic DNA (RAPD). The RAPD are widely used by plant molecular biologists to construct maps because they provide very large number of markers and are very easy to detect by agarose gel electrophoresis. However, the use of RAPD, randomly amplified polymorphic DNA, have certain disadvantages. Firstly, the amplification of a specific sequence is sensitive to PCR condition, including the template concentration, and hence it is difficult to compare between results. The second, another major disadvantages of randomly amplified polymorphic DNA method is that usually it cannot distinguish a heterozygote from one of the two homozygous genotype. The RAPD generation is carried out in the thermocycler in reaction condition similar to PCR. The AFLP, amplified fragment length polymorphism. The amplified length polymorphism AFLP is a diagnostic fingerprinting technique that detects the genomic restriction fragments and is that respects the resembling or the RFLP technique. The major difference between the two steps is that in ALF in amplified fragment length morphism, the PCR used to detect the product in place of southern blotting, as seen in case of restriction. The major difference between the two steps is that in amplified fragments length polymorphism, the PCR is used to detect the product in place of southern blotting, as seen in case of restriction fragment length polymorphism. The amplified fragment length polymorphism approach is particularly powerful because it requires no previous sequence characterization of the target genome. For this reason, it has been widely adopted by plant genetics. However, the same AFLP, alpha amplified fragment length polymorphism, is misnomer, as the process cannot be used to determine variation in length but only presence and absence. The AFLP technique is based on the amplification of subset of genomic restriction fragment using the PCR. 1. Preparation of amplified fragment length polymorphism template is carried out by first isolating genomic DNA, and it is digested simultaneously with two restriction endonucleases, ECO-R1 and MSE1. The first one has 6 base pair recognition site, and the second restriction enzymes has a 4 base pair recognition site. When used together, these enzymes generate a small DNA fragments that will amplify well and or in the optimal size range of less than 1 kb for separation on denaturation polyacrylamide gels. 2. Following heat inactivation of the restriction enzymes, the genomic DNA fragments are ligated to ECO-R1 and MSC1 adapters to generate the template DNA for amplification. These common adapter sequences flanking the variable's genome DNA sequences serves as a primer binding sites on the restriction fragments. 3. Using this strategy, it is possible to amplify many DNA fragments without having prior sequence knowledge. 4. The PCR is performed in two consecutive reactions. In the first, the pre-amplification reactions, the genomic fragments are amplified with with the amplified fragments length polymorphism primers, each having one selective nucleotide. 5. The PCR products of pre-amplified reactions are dilated and used as a template for the selective amplification used to two new AFLP amplified fragment length polymorphism primers which have 2 or 3 selective nucleotide. 6. The ECO-R1 selective primers is radio labeled, and after selective amplification, the PCR products are separated on a gel and the resulting DNA fingerprint detected by autoradiograph. The The amplified fragment length polymorphism technique is of interest as it acts to bridge the gap of knowledge generated by genetic and physical map. The most amplified fragment length polymorphism fragments composed to unique positions on the genome and hence can be utilized as a landmark. In greater plants, the AFLPs may be the most effective ways of generating high density maps. The amplified fragment length polymorphism markers also can be used to detect the corresponding genomic clones. Finally, the technique can be used for fingerprinting clones DNA segments. The methods for detection of cellular functions of genes through protein expression. With a huge amount of genome sequencing being undertaken, it is essential to understand the different proteins expressed in different cell lines. The determination of differential expression of protein across different tissue under different condition is important. Thus, investigation that utilizes different conditions to express and study different sets of protein are being developed. DNA microarrays. The various methodologies developed for the purpose of generating DNA library, PCR, and hybridization have been coupled together to form a basis of DNA microarrays, also known as a DNA chips. The methodology allows rapid and accurate detection of huge number of genes and their products by screening simultaneously. The methodology is composed of 1. The DNA segments from known genes, a few dozen to hundreds of nucleotides long, are amplified by PCR and placed on solid surface like a glass slide. This is done by robotic devices that deposit nanoliters of DNA sample accurately. 2. Many thousands of such spots are deposited in a pre-designed array on the surface area of just a few square centimeters. 3. Alternatively, photolithography can be used to synthesize DNA directly onto the solid surface. This technique for preparing a DNA microarray makes use of nucleotide precursor that are activated by light joining one nucleotide to the next in the photoreactions. A computer is programmed with the oligonucleotide sequences to be synthesized at each point on the solid surface. The surface is washed successively with the solution containing one type of activated nucleotide. As in chemical synthesis of DNA, the activated nucleotide are blocked so that only can be added to chain in each cycle. A screen covering this surface is opened over the areas programmed to achieve the nucleotide, and a flash of light joins the nucleotide to the polymer in the uncovered area. This continues until the required sequence are built up on each spot on the surface. Many polymers with the same sequence are generated on each spot, not just the single polymer shown. 4. After construction of chip is completed, the mRNA and or chromosomal DNA from the tissue or cell culture is added to the chip. 5. The fluorescent chromosomal DNA are often used as a probe, each hybridizing to complementary sequence on the microarray. 7. For example, if labeled nucleotide used to make the chromosomal DNA for each sample fluorescence in two different color, green and red. The chromosomal DNA from the two samples mixed and used to probe the microarray. The spot that fluoresces green represents the mRNA more abundant at the single cell stage. Those that fluoresce red represents the sequence more abundant later in development. The mRNA that are equally abundant at both stages of development fluorescent yellow. The spots that fluorescence provides a snapshots of all the genes provided being expressed in the cells now. For a gene of unknown function, the time and circumstance of its expression can provide important clues about its role in cell wall. The cell protein chip. The protein can also be immobilized on the solid surface and used to help define the presence or absence of other proteins in the sample. The researchers usually prepare an array of antibody to protein by immobilizing them as individual spots on the solid surface. A sample of protein is added, and if the protein that binds any antibodies is present in the sample, it can be detected by fluorescence. Purification of protein complexes. Immunoprecipitation of a protein is of interest can be carried out using the expression of a chromosomal DNA of that protein having an epitope tag. The antibody designed to bind such tag can be utilized to precipitate the protein of interest. If the tagged protein is expressed in cell, other protein that binds to it may be precipitated with it. The identification of associated protein reveals some of the protein-protein interaction of the tagged protein. There are many variations of this process. For example, a crude extract of cell that expresses a similar tagged protein is added to a column containing immobilized antibody. The tagged protein binds to the antibody, and the protein that interacts with the tagged protein are sometimes also retained on the column. The connection between the protein and the tag is cleaved with a specific protease, and the protein complexes are eluted from the column and analyzed. The researchers can use this method to define complex network of interaction within a cell. Isolation, separation, and analysis of carbohydrate. Apart from genes of protein, oligosaccharide of carbohydrate are also of important regarded information transfer within the cell and with the environment. However, analysis of oligosaccharide is complicated by the presence of branches joined by several linkages. The oligosaccharide are removed from their protein or peptide conjugate before analysis. The carbohydrate are subjected to stepwise degradation with a specific reagent that reveals bond position or stereochemistry. The mass spectrometry and NMR spectrophometry have also become invaluable in deciphering oligosaccharide structure. 1. The oligosaccharide groups of glycoprotein or glycolipids are released by purified enzymes like glycosidase that specifically cleaves oxygen or nitrogen linked oligosaccharides or lipases that removes lipid head group. 2. The carbohydrate mixture can be resolved into the individual compounds by some common processes. The fractional precipitation by solvents and ion exchanges and size exclusion chromatography can be used for separation. The highly purified lectins attached covalently to an insoluble support can be used in affinity chromatography to separate carbohydrate. 4. The hydrolysis of oligosaccharides and polysaccharide in presence of strong acids conditions creates a mixture of monosaccharides which can be converted to suitable volatile derivatives. These derivatives may be separated, identified and quantified by gas liquid chromatography. 4. The position of a glycosidic bond is simple. Linear polymers can be determined by treating the intact polysaccharide with methyl iodide in a strong basic medium to convert all free hydroxyl to acid stable methyl ethers, then hydrolyzing the methylated polysaccharide in acid. The only free hydroxyl present in the monosaccharide derivatives or produced are those that were involved in the glycosidic bond. 5. The exoglycosidase of known specificity are utilized to determine the sequence of monosaccharide residues, including branches. The enzymes removes residues one by one from an end. 6. The specificity of this exoglycosidase often allows deduction of the position and stereochemistry of the linkages. The polysaccharides or large oligosaccharides can be treated chemically or with the endoglycosidases to split specific internal glycosidic bonds, producing several smaller, more easily analyzable oligosaccharides. 7. Another major components of carbohydrate analysis is the use of mass spectrometry (MS) and high resolution nuclear magnetic resonance (NMR) spectroscopy. The NMR analysis alone, especially for oligosaccharides of modern size, can yield much information about the sequence, linkage positions, and anomeric carbon configuration on anomeric carbon configuration. 8. The matrix associated laser desorption ionizing mass spectrometry MALDMS is a very sensitive method for determining the mass of entire oligosaccharide chains. Similarly, the tandem mass spectrometry MSMS are readily applicable to polar compounds like oligosaccharides. The tandem MS reveals the mass of the molecular ions and many of its fragments resulting from breakage of glycosidic bonds. A comparison of the masses of each fragments therefore gives information about the sequence of monosaccharide units. Isolation, separation, and analysis of lipids. The lipids are another group of chemical compounds of cellular metabolism that are vastly important for proper functioning of cell. However, extraction of lipid varies from extraction of nucleic acid or proteins, as lipids are non-soluble in water and need to be extracted using a different methodology. Extraction of lipids. 1. The lipid present in the cell membrane are extracted by a polar organic solvents like ethanol or methanol, which reduces the hydrophobic interaction among lipid molecules while also weakening the hydrogen bonds and electrostatic interaction that binds a molecular lipid to membrane protein. 2. The extraction of neutral lipids like triacyl glycerol are carried by solvents that do not allow lipid clustering by hydrophobic interaction. Most commonly used solvents are ethyl ester, chloroform, and benzene. 3. The commonly used extract is a mixture of chloroform, methanol, and water, initially in volume proportions 1:2:0.8 that are miscible, producing a single phase. 4. During the lipid extraction, the tissue is homogenized in this solvent to which more water is added. The mixture is then separated into two phases, with ethanol water forming the top phase and chloroform forming the bottom phase. The lipid are retained in the chloroform layer, while polar molecules such as protein and sugar partition into the methanol and water layer. Separation of lipids based on polarity. The complex mixture of tissue lipids can be fractionated by the chromatographic procedure based on different polarity of each classes of lipid. The chromatographic method have been developed to facilitate the lipid separation based on the difference in polarity. 1. Thin layer chromatography TLC uses an insoluble polar material such as silica gel, a form of silicic acid SiOH4, to separate lipids. In this method, the thin layer of silica gel is separated onto a glass plate. A small sample of lipid dissolved in chloroform is then applied near one end of the plate. The plate is dipped in a shallow container of an organic solvents or solvent mixture, and the entire setup is enclosed within a chamber saturated with the solvent vapor. As the solvent arises on the plate by capillary action, it carries lipid with it. The less polar lipid moves furthest as they have less tendency to bind to the salicylic acid. The separated lipids can be detected by spraying the plate with dyes rhodamine that fluoresces when associated with lipids, or by exposing the plate to iodine fumes. Iodine reacts reversibly with the double bond in fatty acid, such that lipid containing unsaturated fatty acid developed a yellow or brown color. 2. Similar to thin layer chromatography, in absorption chromatography, the silica gel is packed into a glass column, and the lipids mixture is a chloroform solution. In chloroform solution is applied to the top of the column. The polar lipid binds tightly to the polar silicic acid, but the neutral lipid passes directly through the column and emerges in the first chloroform wash. The polar lipid are then eluted in order of increasing polarity by washing the column with solvent of progressively high polarity. The uncharged but polar lipids are eluted with acetone, and very polar or charged lipids are eluted with methanol. Analysis of lipid structure by mass spectrometry. Determining the length of hydrocarbon chain or position of a double bond is carried out by performing mass spectral analysis of lipid or their volatile derivatives. The similar lipids are very much alike with respect to chemical properties of and chromatographic procedure, often cannot distinguish between them. When the effluent from the chromatography column is sampled by mass spectrometry, the component of lipid mixture can simultaneously separated and identified by their unique pattern of fragmentation.